Hope every one of you can see my screen and can hear me out well. So we are going to deal with two important resources during the program. The first one is the edu.t-bio.info. I hope that every one of you have the login credentials for the edu.t-bio.info. Please try it out to log in, and if there is any technical difficulty, please let us know either by writing down on the chat box or you could unmute yourself. So we would be utilizing the edu.t-bio.info for all our courses, projects, and the organization page. So once you log in, you could see that you could see the name of your profile and the profile picture if you have one. Over here, if you hover down, there are courses. Since you are the part of the program, so all the courses would be made available to you. We have the basic courses that starts from Introduction to Bioinformatics, Introduction to Genomics, and a more technical course that is Genomics 1. So you could play around with those courses and try to go through the content of the courses, just so that you have a better understanding about what all is going on in the lectures. Over here we do have the projects. We would be utilizing some of these projects during our lecture, and at the end of the program the participants would be required to work on the independent project. So you could have an idea about the projects and how are they designed by going under the projects tab on the edu site. The other thing that I wanted to show you is the organization page. So what exactly is an organization page? Organization page is a personalized dashboard that has been created for all the participants of the OmicsLogic Genomics program. So if you come down at the bottom, over here you could see organization, and under that you could see it says Genomics training. So that's how you're going to access the organization page. So if you click on that, you would be redirected to a new page, and this is how the organization page is going to look. Over here you could see that we have different tabs: home, forum, progress, curriculum, assignment, and the courses that would be involved. All the important updates about the program or related to the program would be there on this organization page. So for example, um, you're going to have all the events that we are going to cover on the events tab over here, and then the recording, the presentation that is used for the session, and the remote access link. Everything would be available over here on the activity page. So all the important updates would be there posted on the organization page. So it would be nice if you uh, keep yourself updated with all those information. Under the forum, we would have all the recordings and all the presentations or the data set that is being used in the session. Everything you can find it under the forum. You can see that there is one forum that was created by Ilya to welcome all the participants. Other than that, if the participants have anything to share, like a research article or their thoughts, their ideas, feel free to open a new topic under the forum, or if you have any questions you can put it up, and then we or the other participants could answer it in the forum, so that everything is there under the forum. Then moving forward to the progress tab. So for each and every activity that you're going to do on the edu site or on the t-bioinfo platform, we do assign the points for the same, and then you could see the progress of each and every participant, that what are they doing and what are they up to over here under the progress tab. um, so I would like to explain more about the point system. So let's say that if you are on the edu site and if you go ahead and take any of the courses, let's say Introduction to Genomics, so once you register for a course, you would be assigned 400 points. If you complete the course, you would be given 500 points. If you run the pipelines on the t-bioinfo platform, then you would be given some certain points. So that's how we are going to track the progress of each and every participant, and just to make sure that all the participants are moving forward on the same page. So this was the overview about the organization page and the edu side as a whole. Then the second resource that we're going to deal with is the t-bioinfo platform, and the URL is server.t-bio.info. The credentials for the t-bioinfo platform was emailed to all the participants yesterday. So please make sure that all of you are able to log in to the server.t-bio.info. If there is any difficulties, technical difficulties logging in, or if you're not able to log in, please feel free to let us know. So we would be utilizing the t-bioinfo platform for all our practical exercises. It has different sections including NGS data, mass spec, structural biology, and data integration and modeling. If you would like to know more about those sections, you can just hover around on that section, and there would be a pop-up that would let you know what exactly is that section going to cover. Uh, in the platform, we have two different kinds of pipelines. One is the actual pipeline that you would be running when you're working with the real data, and the other one is the demo pipeline. So the demo pipelines will be utilized during the session so that you could play around with those pipelines, and you get a better understanding of each and every algorithm and what kind of an output file you can expect from every algorithm. So I would like to show you both the demo pipeline and the actual pipeline. So if you go under the section of genomics, epigenetics, you could see that there are a couple of pipelines that are listed over here. So if you go ahead and click on mutation variant, over here you're going to see that there's a pop-up that would tell you what exactly is the mutation variant. You could read about it. There's a publication that would let you allow to go in depth about what exactly is the mutation variant, so you can go ahead and read more about it if you're interested. So this is the actual pipeline. I'm not gonna go deeper today because it's an introductory class, so I just wanted to give you an overview. So this is the actual pipeline that you will be utilizing it later in the program. So you have a couple of parameters over here that you need to make the selections from. Once you do the selection, then you can go ahead and start the pipeline. But just to give a more clear understanding about the pipeline, I'm going to go back and I'm going to use the demo pipeline. So if you go under mutation variant and there's a drop down over here, it says demo variant calling. So if I'm going to go and click on this over here, I don't have to worry on any parameters. I can just click on start, and as soon as I'm going to click on start, it's going to give me an overview of what exactly is demo pipeline, what's mutation, what is this pipeline going to do, and how you're going to analyze the genomic data. So we do try to educate and reiterate at each and every level. So whatever algorithm you're going to select, there would be a pop-up that would give you a brief idea about what that algorithm does and what's the input and the output file that you can expect. So if I'm going to click OK, after clicking start, I could see that there are several highlighted buttons. That those buttons are the algorithms that you could utilize. So let's say that I have a genomic data and I want to do the pre-processing of my data. So how would I be able to do the pre-processing? Over here you could see there's a section that says data pre-processing. So this section is dedicated for pre-processing of my data. So in this, I'm going to select PCR clean. So it would give me an information about what exactly is PCR clean. I'm going to click on OK. And then there's an option to map. So I can map using bowtie 2. I'm going to click on OK over here. It's going to give me what exactly is bowtie and the publication if you would like to read more about bowtie. I'm going to click on OK, and then I'm going to see if I would like to use something else. Over here, I have an option under differential variant calling to use the algorithm Strelka. So we can go ahead and click on this. So once you go back to the edu site and you're going to take the courses of Introduction to Genomics and Genomics 1, you're going to have a better understanding about what all these algorithms do and what's the significance of these algorithms. So that's how you're going to create your own pipeline, and then once you're done with the pipeline, you're going to click on end, and then that's where you're going to get the output files. So that's the logic behind having the demo pipeline, so that you could play around with it. You don't have to worry about the input files. Everything is there. It's just for the practice purposes, and you don't have to wait for the pipelines to get over with the demo pipelines. So those are some of the benefits of the demo pipelines. The last thing that I would like to mention is, on the edu side, you do have an option to update your profile. If you click under their name on the top border, there's an option that says profile. It's very important if you could complete your profile. And what I mean with completing the profile is to have a good profile picture, to have social media links, and some background information about you, so that the participants can know who is the other person and with whom are they having this program, so that they can reach out to you, they can talk about projects, they can talk about collaborations. So the idea is to build a close community where you could discuss all the opportunities, all the ideas related to bioinformatics, related to genomics data analysis, and things like that. So I would like to show you some of the examples of the good profiles, I would say. The first one I would like to highlight is Kun. You could see that she has a good profile over here. She has around 11,000 points. And as I mentioned initially, that for each and every activity that you do on edu site and on the t-bioinfo platform, you're going to get the points for. And that's why it says over here that she has 11,000 points. She has a profile picture over here. And if you go under yeah, and you could see that all the activities are listed over here, that she did start the pipeline, and then she finished a specific pipeline, and then all the participants of the OmicsLogic program would have a badge that is assigned to them. So that means, like, you're the part of that specific organization. So Kun is the part of the precision oncology program, and that's why she has this program. We do have a good profile from one of our participant today in OmicsLogic. It's for Amit. So you could see that he has a profile picture, he has a badge, and he did try running some of the pipelines. So these are some of the examples of a good profile. So it would be nice if you can go back and complete your profile. And in the last, I would like to highlight the profile for Despina. So we could see that she has all of the information about her, what she has been doing, what's the position, her social media link. She has 19,000 points, and then she is the part of the organization OmicsLogic, and that's why she has the badge over here. And then you can directly go out and check her LinkedIn profile as well. So now I would like to ask Despina to come on the stage and introduce her and say a few words about her.
Hello, everyone. My name is Despina Kalfakakou. First of all, I'd like to welcome all of you to the OmicsLogic Genomic program. I have to say that it is a great pleasure for me to guide you through the program. And okay, let me tell you a little bit about the program. So for the next weeks, we will go through some basic and more advanced concepts of genomics data analysis. By the end of the course, you will have gained the necessary knowledge and resources to design your own analysis of genomics data. But this all will be presented to you afterwards. And let me tell you some things about my background. Excuse me? Is someone talking? I can't hear you.
Let us redo the whole thing that was done, right? And that it cannot be quickly done. It takes time to. That's where I'm struggling.
So if someone is talking, I can't hear you.
While Despina is speaking, if someone is unmuting, I mean, can you just mute your microphone so that Despina can speak? Thank you.
Okay. So, okay, I am from Greece. I have a background in computer science. I graduated from the Department of Computer Science at the University of Athens. I continued my studies for my master's degree in bioinformatics at the same department. And since 2016, I am the bioinformatician of Molecular Diagnostics Laboratory at Demokritos. Demokritos is the largest scientific research center in Greece. So in our lab, Molecular Diagnostics Labs, we study hereditary cancer. Molecular Diagnostics Lab is a reference lab in Greece for hereditary cancer. We have been studying hereditary cancer since 1998, at first by studying specific genes that were known to predispose to hereditary cancer syndromes, while since 2014, with the help of next generation sequencing technology, we are studying more genetic loci, which we don't know yet if they are, if mutations in these loci are responsible for hereditary cancer. So in the lab, I am the only bioinformatician among geneticists, and I am responsible for all the bioinformatics pipelines and analysis performed. Our goal is to decipher the mutational spectrum of hereditary cancer in Greece. And up until now, we have analyzed more than 10,000 cancer patient families, while almost 6,000 of them are analyzed by next-generation sequencing. So in parallel, I am conducting my PhD studies at the University of Thessaloniki. And my goal, my PhD's goal, is to develop bioinformatics tools and algorithms in order to analyze data and identify genetic biomarkers that will diagnose hereditary cancer and maybe help patients benefit from targeted therapies. So for the next week, me with Pratim and Sahil that you see in the screen and Elia, we will guide you through genomic data analysis. And I hope that we will all have fun and learn many things. Thank you.
Thank you very much, Despina and Sahil. And hope everybody is appreciating the effort that we're putting in to make this program successful for you. And I briefly wanted to touch on the point of what might be the outcome, why you would benefit from going through the online materials and through the practical aspects of the program. And again, review some of the topics that we will be covering. At any point in time, feel free to add your questions or comments in the chat. We do want to hear your opinions or any questions and make sure that we address those in a timely way. We have a chat box, as you have seen, both the platform and on the educational site, and you please can feel free to communicate with us. Sometimes we will ask you what is your email ID and what is your name, and that is just for us to help connect your question with your account and see you know, what we can add to the program, if anything. So I do want to just also add that we are recording these sessions. Every time after the session, after several maybe hours, we will be posting the recorded version of this webinar on the forum. So don't worry, you will get the recording. If you miss one of these sessions, you will still get the recording, and all of that information is going to be available on that program page that Sahil already showed you. So as you see, we have several people here. We have Sahil, who will be dealing primarily with the organizational aspect of the program. Despina is really the bioinformatics expert that has the experience in analysis of genomic data, and she will be guiding you to understand the theoretical aspects of some of these analysis methods and giving you some of those examples. And Pratim, who also has a lot of experience in bioinformatics and is at the same time teaching several other programs that we are conducting, he will be here to answer any questions, facilitate the discussion, and help provide additional materials, publications, or other materials that would be helpful for you to appreciate the way that genomic data is making a difference in cancer and a variety of other conditions that we will be talking about in this program. So what is genomics in the context of this OmicsLogic program? The OmicsLogic program was established to cover 4 main areas of omics that we have identified as a primary interest for a lot of the people that we have been interacting with, and that are critical for the understanding of the way that molecular data is impacting a variety of areas, from infectious disease to oncology to agriculture and to neuroscience and other areas where research is producing a lot of interesting discoveries, and a lot of those discoveries are also translational to the field of medicine, biotechnology, pharmaceutical industries, etc. So the OmicsLogic program covers the area of genomic data, transcriptomic data, epigenomic data, and metagenomic data. And typically the way that these programs are structured, they're anywhere between 2 and 3 months, depending on the type of audience. And we have already been testing this program in a variety of universities at different levels, at the undergraduate level, at the graduate level, and of course we work with a lot of industry professionals as well as researchers. The goal of the program is to empower you to do your own research, be able to analyze your own data, and have the theoretical understanding of both the methods, the type of data, and the applications of this data in a variety of industry and research applications. So as we start off with very basic ideas to refresh our memory about what is DNA, what is DNA code, how can we analyze that type of information, we are really trying to help facilitate a process where you are familiar with the type of information that you might be dealing with. You can start asking questions of that data and eventually develop a whole project, either using data that you already have, that you have generated yourself in your lab or your collaborators, or using publicly available public domain data, which we will also show you where you can find and how you can download and help you upload it to the platform, so that if you do not have the infrastructure to analyze some of these larger data sets, you will be able to do that as well. So again, the goal is to give the theoretical background, but fairly quickly to jump into the practical aspect, allow you to work independently. And if you are not ready to work on a project independently, help you find another project that maybe somebody else is working on, so that you can get plugged into that project and get something practical out of this program. We are conducting a number of sessions that will be much more focused on various aspects of genomics. You might have questions related to some algorithm that we haven't covered, or you might have a specific question about a project. Obviously, there's a variety of resources that we can point you to or help you find so that we can answer those questions. But let us go through quickly the topics that we found to be important to cover in this program, just one by one, and kind of explain a little bit how this will be structured. So first of all, I want to say that we have scheduled approximately two meetings per week. So I will be going through some of these, but of course this is not a comprehensive list. And then at the end of this, I will show you where you can find all of this material. We have a previous recording where you can see the topics that we will cover, that we covered in the free webinar. And we also have all of this information on the Genomics program tab that Sahil just showed recently. So we will start today, introduction to the program. We kind of gave you a program overview of tools and resources. Some of the expectations, again, the expectation is that all of you will feel comfortable enough to analyze your own data. And now we will go through the materials, and you already got introduced to the speakers and the presenters in the program. And so we hope that this will be a two-way conversation, an interactive session, as well as a lecture that you can benefit from. So first of all, we will talk about DNA and the code that DNA has. What are these letters? Different genomes that are out there. We will be focused a lot on the human genome, but not only. We will also look at genomes of, for example, viruses or bacteria. And we will talk about the different types of genomes and what do we need to keep in mind as we look at these different genomic data. So we will also look in detail at the different technologies, sequencing technologies, that have been developed over time. And primarily, we will focus on the next generation sequencing technology that produces short reads. And those short reads have to be mapped on that reference genome to be able to identify which part of the genome did they really come from. So we will talk about the DNA structure, the variants, the chromosomes, and then we will also talk about how these different sequencing methods help us identify variants and see what might be the significance of those variants. Then we will start talking about processing this data. And so when we say processing genomic data, what we mean is structuring that data. Because first of all, we get access to raw, large data sets that really are difficult to sift through if we want to identify something of significance for our analysis. So what we will cover there, again, are the different types of sequencing, and that's important because that gives us an idea into what kind of information we will be able to find, but also some technical aspects like the read lengths, the mapping, different mapping techniques, and how to select the right reference genome, of course. And what we will see is that there are actually many different techniques that utilize the same next generation sequencing technique, but really are designed to show us different type of information. So what you can see here, again, is a chart. Over the years, you can see an explosion of methods and short sequence aligners for DNA, RNA, methylation, microRNA data. So a variety of data types, a variety of different types of information that we can extract from this. And the basic idea that we will first focus on is how do we align this data to the reference genome, and then how do we visualize it? Now, any type of FASTQ data you can put into an aligner, and it will align it to the reference genome. And we'll talk a little bit about the different types of sequencing, also some of the ones that you are mentioning in the chat, PacBio, Oxford Nanopore, so different types of sequencing that are not traditional next-generation sequencing like Illumina does. But we will talk about these different sequencing techniques in the context of how does that impact our ability to align them to the reference genome and then visualize the results. And then we'll briefly talk about the overall idea behind processing of this data. So for example, we first do pre-processing, eliminate some additional adapters or amplification of reads, and what would be the impact of pre-processing on specifically genomic data. We'll talk about the different techniques for mapping. Visualization is a step that is helpful but not critical for the analysis itself, but we will go into that aspect as well. We'll talk about the variant calling and how we utilize statistical approaches to differentiate between noise and the different types of variants. And then we'll jump into the analysis, for example, differential analysis or other type of analysis. And finally, annotation. How do we annotate? And all of that is encapsulated in the platform. And the goal is to really help you jump into the analysis quickly and not spend the time to install all the different packages and set up your environment first to learn how does it work, and then if you desire to do that on your own, we will be happy to assist you with that as well. So then we'll jump into additional aspects of the processing, and I do want to just show you a quick example of this pipeline. So Sahil already showed this, but just quickly to give you an overview, really the goal is to eliminate any kind of technical difficulty, and as you build your pipeline, to learn about the impact of your different selections that you are doing, and then quickly see the result. What is the output? And then how can we evaluate the output of multiple types of techniques and really see what would be the result of that analysis. So that is really the goal of this initial step, to introduce everybody into the access the data, having to go through several steps to get that data to be useful for analysis. And once we understand that, then what we will go into are the different types of DNA variation and, of course, the different methods that we would need to use to identify those ways the DNA could vary from, for example, the reference genome or from a normal sample that is from the same organism. So we'll talk about different types of mutations. For example, germline mutations, somatic mutations like the ones that arise in cancer as a result of cell division, and we'll talk about other non-point variations, so copy number variation. So what we felt was important for us is to give a broad overview of a variety of different DNA variation types, but then to go closer and look at how those types could be interpreted in the context of a project. So we'll spend a little bit more time. We actually have several parts for some of these bigger topics. And specifically, when we talk about DNA variation, we'll see how the different types of variants, we'll specifically focus a little more time spent on the single point variations. And we'll talk about how different types affect, essentially in a different way, the product of whatever is encoded in the DNA. And as a result of our ability to look at the different types of variants, we will look at how it's important to understand both the biology of DNA code, transcription of that DNA code into RNA, and then translation of that RNA into protein, the central dogma of biology, but also we'll look at how just technically these variants have an impact on what we are trying to analyze. And so we will use visualization, including visualization in JBrowse, that is a part of the platform, as well as other visualization techniques where we can start incorporating more information into what we are seeing. For example, when we map our reads and we identify genes that are there in the GTF file, how can we add additional information about the variants and the common, for example, single nucleotide polymorphism database that will give us an idea of what is known about a specific position. Or how we can use reference studies, for example, the Cancer Genome Atlas, TCGA, to understand in what other cases has a specific mutation been studied and associated with a specific outcome. So that will bring us to the point of being able to analyze some of these genomic variants. Because if you think of the length of the DNA, the 3 billion base pairs, and you think of the amount of variants, which is typically we think of about 1% of our DNA having significant variation, 1% of 3 billion is a lot of variants. So there is a lot of room for us to get stuck in just a lot of data, and we don't know what to do with a lot of that data. So we will talk about how we can, for example, filter some variants and focus on a specific type of variant or a specific selection of variants. We'll talk about how we can leverage the coverage. And when we talk about some of these different sequencing techniques, when we talk about whole genome sequencing, whole exome sequencing, targeted sequencing, some of these long read methods like PacBio or low quality like Oxford Nanopore, how does the impact of the error rate and the impact of the length of the read, the coverage of the read, how do all of those aspects factor in to our ability to analyze in a statistical way the variants that we have identified for significance and use that to understand the difference between our samples. So we'll talk about that again. We'll spend some more time. Actually, a couple of meetings will be dedicated to that. And we will talk about that these variants, they essentially affect and help categorize the mutations in terms of what they would mean. So when we talk about that, we'll talk about missense mutations, nonsense mutations, frameshift mutations, and we'll see how these ways of categorizing mutations are actually going to help us focus on a specific type of variants that we are going to be discussing in the context of a project. As I already mentioned, we can now use this information to annotate. And now we can understand what's in our data and try to bring it closer to more of a biological understanding of what's going on. For example, we can start looking at specific variants that are affecting the genes in a specific way, and then abundance of those mutations associated with a specific type of a condition. How do we leverage all of this information of being able to filter the variants, being able to focus on a specific type, being able to then leverage databases and reference studies and tie all of that together? And sometimes we will look at reference databases as maybe one, so to say, supervised direction, and we will look at more association studies, like for example genome-wide association studies when we have a phenotype and a genotype and we need to understand the relationship between them. So all of these things will be covered first in the context of the platform, because the platform really all that it does is it puts the different methods together in a way that we can all follow along and produce a result. And then in each method, as you have seen Sahil has pointed out, each method is described in detail, a lot of times with the code that you can download, and we'll show you some examples of that, and then you'll be able to set up that pipeline independently. But of course, that is not the focus of the course itself. The course is not going to be training you how to set up the pipeline. It's going to be focused on understanding the methods, understanding the types of challenges and the opportunities that genomic data provides, and being able to leverage that to work independently on your project. So, but we can talk about that if you're interested in the aspect of technical understanding of the methods and how to make them work. We can talk about that as well. Of course, somebody like Despina in her lab has to regularly set up pipelines to process and annotate some of the data that they're working with. So I'm sure that she will be able to speak to that from her experience. Now, we will also look at some of these repositories and databases that are regularly used in the analysis of specific types of mutation data. So for example, as you can understand, the types of reference databases and studies for cancer are going to be very different from, let's say, infectious diseases or, rhizobacteria from the roots of plants. So there are going to be a variety of different types of databases and resources that we will cover, and we will talk about how do you find a data set that you will be able to use in the context of your project, and how do you then validate what is the validity of some database and how reliable is that source. So we'll talk about some of those aspects, and then we'll talk about pure databases that just have all of this information collected in a specific way that you can query, or simply a collection of data sets that we can use as reference. And so we'll talk about some of that. And the way we would like to do this is really tie this to the project that you are working on. So really, if you are starting to think of a data set, you're starting to think of a project that you have been working on, and you are starting to formulate your question in terms of the types of methods that we have covered, the sooner we can start having that conversation, the quicker we can get that process started, and hopefully all of us will benefit from working in a real use case scenario and not just theoretical knowledge that becomes very impractical when you face specific challenges. An important aspect of analysis is not just the annotation and the filtering, which is a standard approach, but also using statistical methods, machine learning methods that help us identify relationships in complex data sets. So when we talk about specifically genomic data, we are meaning a very specific type of data. It's different, for example, from transcriptomic data or from metagenomic data. And a lot of our ability to analyze this type of data in a non-linear fashion, in a fashion that really incorporates the complexity, is first preparing that data in a very specific way. So up until this point, we have been dealing with processing or preparing that data for our ability to analyze it. And as we said, that will include the pre-processing steps, the mapping, the annotation, for example, annotation by gene or annotation by type, and then preparing essentially a table. But once you have that table, then you're able to deploy some of these more complex machine learning techniques that could be supervised or unsupervised. And a lot of these techniques are now being used in profiling of patients, for example, stratification of patients, identification of signatures and associating those signatures, so selecting specific positions or specific types of mutations or specific levels of mutation abundance with a specific phenotype. And we'll look at how this is used in biomarkers, as well as some other use cases, for example, in the development of vaccines for infectious diseases. So really a lot of different exciting applications of machine learning for genomic data. But of course, because it's genomic data, we have to be very specific about how do we prepare the data and what does it look like. And then this will also guide our ability to understand the machine learning output and interpretation of the results. So again, because this is a complicated topic and it requires some understanding and some practice, we will actually spend a whole 3 sessions to talk about machine learning. And some of the things that we will be talking about are, for example, unsupervised techniques like principal component analysis that can very nicely summarize the data about samples based on genomic variants and based on other aspects, for example, abundance of mutation in a whole genome. So we'll talk about some of these techniques, and we'll talk about how these techniques are used in a specific project. As an example, as you can see here, we see a timeline of Ebola, the outbreak of Ebola. As you can remember, the previous outbreak of Ebola in 2014, 2016 was a very interesting and, of course, dangerous event that happened and forced us to understand, try to understand how the genomes of these individual viruses developed over time and how can we associate them with the location, with the virulence, and with the outcome, how lethal, how pathogenesis of that virus changed because of those mutations. And now, because we were able to utilize sequencing in real time, sequencing data from patients, taking samples from patients and culturing them, seeing how these techniques can really inform our ability to intervene, our ability, for example, the vaccine, the Ebola vaccine that is now being used in Africa in the current outbreak was developed as a result of that previous outbreak. So we'll look at that example project, we'll look at machine learning techniques in the context of that project, and as you can see here, machine learning, of course, has a variety of applications and a variety of topics that will be covered inside of this broad kind of a topic of machine learning. Finally, we will transition to some of the projects. And the projects are of three kinds. One kind are the projects that we have prepared. And so some of the examples you can see here: cancer studies, where we will look at hereditary mutations that are contributing to the risk level of how much risk is there for a patient to develop a specific cancer. And we'll look at somatic mutations, how we can detect early onset of cancer, or, for example, understand the staging, understand the type, etc. We'll talk about tuberculosis, which of course is another interesting area for many groups around the world studying MTB specifically, where there's a resistance to different treatments. And we'll try and look at how we can detect resistance in microbial infections and how we can associate them, for example, with the source, where they came from. And this is, of course, an important topic, and we'll kind of touch on that in this course. We'll talk about case control studies and genome-wide association studies. And also we'll talk, as I mentioned, about other types of non-human diseases or non-human genomes. So some of these examples are right here: the somatic mutations in cancer, the drug resistance in tuberculosis, viral adaptation in Ebola, and an example with genome-wide association studies using rhizobacteria, but also some other examples as well. And so we do want you to know that, you know, the reason we are starting this program now in the summer, and the reason that we are excited about this, is also because we are doing this specific program for the first time. We have done it in the context of other types of programs where genomics was only a component and not the full program. But this specific genomics program, we are doing it for the first time. And I really want to say that we appreciate a lot the work that Despina, Pratim, Sahil, and others have put into this program. We are still finalizing some final touches and preparing some materials. So please keep that in mind. But, you know, we do think that we have some great content that is ready to be shared with you. And we are excited to see the projects that you have. And of course, a lot of these projects, they are research projects, right? And so research projects do not always yield concrete answers or yield very clear results. And there's always an aspect of being both an understanding. We have to come to these projects with an understanding of what we are trying to get out of a project, what is exactly our question. We have to come at it from an understanding of the tools that we're using, how these tools work, what are the parameters that we can adjust for these tools to work properly, how do we combine these tools together to give us the answer to the question that we are looking for, and then finally, how to interpret. What do we do with the results of these methods? How do we look deeper than just what first appears on the surface? How can we also be critical of the results of the analysis? And how can we ask ourselves whether the results are not only satisfactory for us to give us a broad idea of what's going on, but really how do they answer the question that we first originally posed? So we'll talk a lot about these aspects of research in these projects, but also we will talk about some of the exciting translational value of these projects. For example, we saw that Despina's lab just in June had a publication come out, and have some results from the analysis of the data that they have. And we'll talk about how it's important to generate some of these hypotheses, maybe utilizing public domain data, but then a lot of the ability to publish and to have these conclusive studies is also the data that you have access to. So we'll talk about some of the significance of the data types, the data sources, the amount of data that you should have in a study that is impactful and conclusive. So again, those are some of the aspects of the program, the schedule, the topics that we will cover, the project examples, and of course you are not limited to the projects that we are preparing for this program. You are also welcome to work on your own projects, come with your own data sets, or leverage some of these public domain repositories where we will show you how to find data that might be relevant to the topic that you're studying. So I do want to, again, highlight the people that will be your primary connections. Despina, who's really an expert in the clinical genomics and bioinformatics from a computational side point. Pratim, who has a lot of experience and is doing right now a lot of program planning and interaction with all of you. Sahil, who knows the platform and knows the educational resources that we have. And of course, I'll be joining this program as well. At this point, I really want to spend the next several minutes that we have left for questions and answers. And I welcome Pratim, Despina, Sahil, any of you to add to what I have covered so far. And any of you that are a part of this program, we want to mention that we're excited to see what you can produce during the program. And if you have any questions or if you have any comments on what we've had so far, please either add them on chat or you can unmute yourself and speak, and we'd be happy to answer any of that.